Okay, guys, we're going to start chapter 10, muscle tissue. First thing, talk about some of the characteristics of muscles. Muscle tissue is excitable. That is, it is dependent upon neural stimulation in order for it to contract. It does not do it on its own. Now, there's different neural stimulations for these three types, the skeletal, the smooth, and the cardiac, but they all are excitable. Elasticity, the muscles uh, can be stretched, and that actually adds to the amount of force that they can generate. Extensibility, again, it's stretching, and this is the part that really defines muscle tissues, contractility. So if I had something right here, and I had something right here, and I had a thing in the middle, if this thing in the middle could pull these guys toward the center, that's a muscle. That's exactly what happens. All muscles will pull. There is no such thing as a pushing muscle. Well, how come I can push? It depends. One muscle is pulling in the opposite direction. That's what's causing the push. But all muscles have to pull. They have to contract. They cannot push. Okay, as far as the connective tissues, again, if you've had or if you're in the lab, this is a repeat. That's fine. Doesn't hurt to repeat it. So we have this connective tissue sheath that surrounds the entire muscle, and it's called the epimysium. It's dense, irregular connective tissue. Now, the major divisions of a muscle are called fascicles. They're a group of several muscle cells or muscle fibers, same thing, that are grouped together, and they're surrounded by their own connective tissue sheath, and it's called a perimysium. And finally, each individual muscle cell, which is called a muscle fiber, is surrounded by a connective tissue sheath that's called the endomysium. Endomysium, collagen, reticular fibers, and then, of course, underneath that is the plasma membrane, but in the muscle we call it the sarcolemma. Okay, guys, next slide, muscle fibers. Let's kind of break this up a little bit. So there's our muscle fiber or muscle cell, single cell. Yeah, there's multinuclei. That this is obviously would be a skeletal muscle cell. And making up each of inside that one muscle cell are all these myofibrils. The myofibrils are going to contain the actual contractile structure that makes a muscle pull. Obviously, lots of mitochondria because muscle needs lots of energy. Okay. Then we have the sarcomeres. Sarcomeres are the subunits of the myofibril. This is the most basic contractile unit you have. I'm not going to draw on that. I'll draw over here. But basically, it mirrors what I showed you guys on the previous slide. Only these guys, these zigzags, are called Z-disc. And that's what these thin filaments are connected to. In the center, we have an M line, which is kind of like a disc as well. And this is what the thick filament, try to draw this a little thicker, guys, is connected to. So, seeing here, there's the Z-disc. Between one Z-disc and another Z-disc is a single sarcomere. Okay, these green lines are representing the thin filaments. Thin filament is also made out of the protein actin. And the thick filaments are represented by these purplish lines, these thick ones there. And of course, that is representing the thick filament, or myosin. Okay, now as far as all these bands in this H zone, the I band, if you look, the I band goes from the end of a thick filament in this sarcomere to the beginning of the thick filament in this one. So basically what the I band is a place where you have no thick filaments. And on here, you can see up here on this one, that's the lighter part of the muscles. You guys may remember when we were doing the tissues, I told you I would tell you later why muscle is striped. And well, this is later and I'm telling you. The I band is the lighter because there are no thick filaments in there. Okay, the A band is from one end of the thick filament to the other of a single sarcomere. So that's going to be an area where you're going to have the thick filaments. Now, in this A band, you also have part of the A band is going to have the thin filaments in it as well. So that A band is going to be the darker stripes that you see on the muscle fibers. The H zone is an area where all you have is the thick filament. There is no actin. The thin filament is outside the H zone. Now, here's something else, let me tell you. The H zone actually goes away when the thick filament pulls the thin filaments toward the center from both sides. Eventually, the H zone gets smaller and smaller and goes away. Alright guys, that is the ultra anatomy of the muscle. Here's another view of it. Same thing, Z-disc. You see the thin filaments, you see the thick filaments, the M-line, the H zone. The H zone is a little bit, oh, there's the H zone. Okay, I was about to say that H zone was too wide. Now they're just showing you the sarcomere, which is from Z-line to Z-line. We see the A band in here, which is the area where we have the thick filaments of a single sarcomere. We see the I band, which is from this thick filament to this one. It actually covers more than one sarcomere. But the main characteristic of this guy is there's no thick filaments in there. You can see the M line, which is what the thick filaments are connected to. Now, let's look at the thick and thin filaments. This is what the thick filament looks like. It has these things that look a lot like a golf club head. And these guys have areas that bind to the actin. They also have an area, of course, to bind ATP. So there's the head and the hinge and the tail of the myosin. Here's the actin. And if you look at it, it's two strands that are kind of twisted around each other. Now, on that actin, you have these guys right here, and these guys are called troponin. Troponin is going to be the thing that's going to keep this long piece right there, which is called tropomyosin. What tropomyosin does is it blocks those binding sites right there for this guy. So as long as tropomyosin is there, then these guys can't bind to it and nothing can happen. No muscle movement. Now, when calcium binds to the troponin here, then the troponin actually changes shape and it will actually move the tropomyosin off away from these binding sites, and at that point the myosin can do its thing. Titin is going to be one of these things that help us to connect the actin to the Z line. Okay, neuromuscular junction. We said that there is no way for a muscle to move or to contract without neural input. The neuromuscular junction is this whole area right here. What it basically includes is the end of the nerve and the surface area of the muscle cell that has receptors for the neurotransmitter. For skeletal muscle, it's going to be acetylcholine. That's going to be what's going to be released from the end of the synaptic bulb of the neuron, the nerve cell. It's going to float across this gap, which is called the synaptic cleft, and it's going to bind to these receptors. You can see them right here a little bit better. It's going to bind there. Of course, that's the sarcolemma, which we know is just the plasma membrane. The nerve impulse travels down the axon, and then when it gets to the end, it opens up calcium gates. Calcium floods in here, and when calcium floods in, these guys exocytose out and then float across that synaptic cleft to the acetylcholine receptor. And by the way, this part of the muscle cell is called the motor end plate, because this is where all the action is going to be taking place. When acetylcholine binds to this guy, he opens up, and as you can see, he allows sodium to flood in. That's his whole point. When the sodium comes in, remember that the inside of the cell is usually negative. Not exactly, but here minus 70 millivolts is good enough. 50 to minus 100 millivolts for cells. And when the sodium comes in, it's going to change that, because all the positive charges are going to change it to a plus 30. When this changes, it's going to cause some other gates here. These are sodium gates too, but these sodium gates don't have receptors for acetylcholine. These guys are going to open when the voltage changes. So these are voltage gated sodium gates. So the plasma membrane or sarcolemma of this guy actually evaginates into this little T-tubule. This T-tubule is running adjacent to these guys. Oops, didn't make my X there. These are the terminal cisternae. Those are the ends of the sarcoplasmic reticulum. That sounds almost like endoplasmic reticulum. That's what it is. In the muscle cell, they call it a sarcoplasmic reticulum. These terminal cisternae store calcium. That's what these guys do. So when the voltage, those voltage gated sodium channels here open, we get this change in voltage. It's going to cause the terminal cisternae to open their calcium gates and to flood the inside of the cell with calcium. Who cares about that? Well, it's part of what we need. So we see the action potential, which is the electrical signal traveling down the end of the nerve, gets to the bulb. We already said calcium gates open here. These guys do exocytosis. They're going to release, they're going to bind to these ligand-gated sodium channels. They're going to open, let sodium flood in, and of course, then this is going to cause the voltage-gated sodium channels to open up as the voltage changes, and that's what's going to cause the SR, which is right here, is going to cause this guy to release the calcium. Now, remember I told you that's what binds to troponin. When the calcium binds to troponin, tropomyosin moves away and these myosin heads can make contact. What difference does that make? Well, these guys are going to make contact and they're going to pull this guy toward the center. Remember we said that's how the muscle works. So the sarcomere is going to get shorter. All of these sarcomeres in this myofibril are going to get shorter. All of the myofibrils in this muscle fiber is going to get shorter, and then a variable number of the muscle fibers are going to get shorter and basically make the entire muscle get shorter. And when it gets shorter, these guys, remember, are attached to bones, and they're going to pull on the bone and make you have a movement. Okay, once the action potential is over, this acetylcholine is going to be released from the receptors. It's either going to be taken back up here or it's going to be deactivated by a chemical that is floating around called acetylcholine esterase. But once it's over, calcium is going to be reabsorbed back into the SR, and once it dissociates from the troponin, the tropomyosin will be caused to cover the actin binding sites. Basically, the muscle is going to relax and get longer. Just another version of it. Now, here, again, here's this H zone right there. The muscle contracts. What happens to the H zone? It gets too super tiny. It basically almost goes away. The actin can actually make contact with the M disc or the M line. So another version of looking at it. Okay. Let's talk about this actual contraction. So there's our thick filament. The calcium is bound to the troponin, so the binding sites are unblocked. The myosin is going to bind to it. Now notice that this guy has ATP that's already been broken down into ADP and a phosphate. He's going to, that's the cross bridge, by the way, when it binds there. When he's going to release that ADP and that phosphate, and when he does that, that's the power stroke. In this case, this guy is pulling the actin this way. Okay? Now, he's going to release from the actin once another ATP binds to him. Okay? ATP causes him to let go of the actin. Now, he's going to break it apart into ADP and the phosphate. And when he does that, this is the cocking mechanism. This guy is cocked and ready to go back up here and start again. So that's how we can move that actin toward the center. Where do we get ATP from? Well, we get most of it from the mitochondria. We get a lot of ATP from that. We use blood sugar. We can use a pyruvate. We can use fatty acids. We send it through the Krebs cycle and goes through the mitochondria, and out comes a lot of ATP. We can also do glycolysis. We do this when we can't get any oxygen. We take our blood sugar and this glucose, and we make 2 ATPs and 2 molecules of pyruvate. If there's no oxygen, then that pyruvate gets turned into lactate or lactic acid. If it has oxygen, then we just hand it back off to the mitochondria and finish up. Now there's also, hopefully you guys remember, but if not, I'll refresh it. We take our excess glucose and we make glycogen in the liver, but we also make glycogen in the muscles. So the muscles have an onboard, not a very big supply, but a supply of blood glucose that they can use themselves. Okay, so there's glycolysis and aerobic respiration. That's how we're going to get the ATP. Now, here's yet another thing. We can take ATP and creatine and we make it into creatine phosphate. We come up with an ADP, and this creatine kinase is going to be able to take that ADP and an inorganic phosphate group and make it into ATP and turn it back into creatine, which is right here. So this is just a third pathway to make ATP that the muscles can do themselves. Types of contractions. That man has got high hopes, and I don't see it, but we have two basic types of contractions. We've got isotonic and isometric. Isotonic contraction is this one right here. This guy has got a dumbbell, and when he contracts his muscle, it moves. So what happens? The muscle gets shorter. Look how long the muscle is, and look how short it gets. So he moves it. He contracts his muscle, and the muscle gets shorter. Now he can do that coming down this way. He can also do this coming down as well as coming up. In this case, the muscle, this would be like when you go back the other way like this, but you don't just drop it, you take it a controlled drop. You are slowly allowing the muscle to relax, but you're also keeping contraction so that this thing doesn't just drop down fast. It goes at a controlled rate. So both of these are isotonic. Isometric, this is isometric. This guy is pushing as hard as he can, but he's not going to be able to do it. Isometric, you contract your muscle, but say this had too much weight on it, you can't move it. The muscle contracts, but it gets no shorter, does not shorten or lengthen. The amount of force is less than what is needed to move things. So this is the concentric. When we shorten a muscle, the eccentric is when we lengthen it in a controlled manner. Isometric, no movement at all. Motor unit, we talked about the motor end plate. A motor unit is a single nerve, a motor neuron, and all the cells that it controls. That, even though there's only one axon on a nerve cell, it splits into different ends. There can be several of this. Small motor units is where a neuron has a very small number of muscle cells. And this would be things that you have extremely fine control over. Your eye muscles, the extraocular eye muscles that move your eye, and the fibers in your, or the nerves and muscle fibers in your fingers, you have fine control over. And these guys, basically one neuron only innervates 6 muscle cells. Large motor units, such as the one in the big thigh muscles, well, a single neuron does hundreds or thousands of muscle fibers. These guys, they're not required to do any kind of fine movements. All they have to do is do a big contraction. Now here's a concept we need to go into: recruitment. Say you reach out to pick up something, and you reach out and you think that the object you're going to pick up is light, but when you try to move it, you find out it's much heavier than you thought. But you still are able to move it. So what's happened there? Why didn't you move it the very first time? Well, since we do have motor units, I don't have to use every muscle fiber in a muscle. I can use only part of them, which is why we can crush an aluminum can in our hand or also hold a little baby bird or something. We have the ability to control the amount of strength that our muscle exerts to a degree, and it depends on the muscle, whether it's small or large. But recruitment is where if you reach out to do something and find out that the amount of force you're exerting is not enough, you can call on additional motor units by getting more and more of the muscle cells involved until you have enough to affect the action that you're attempting to do. Okay. Alright guys, this is a graph showing length-tension relationship. I told you a few minutes ago that you can actually generate more force if you stretch your muscles to a degree. You can, and I'm about to explain to you how that works. So, say this is our, let's come down here. This is our actin, and this is our myosin heads. Now, if I stretch these like this, then I can generate more force because I have further that I can move these guys. That's what that's saying. By stretching it, increasing the length right there, you come to a point where you actually are able to generate more force. Obviously, if the actin were like this, I don't have very far that I can actually move that, so I'm not going to be able to generate very much force. So decreased length is going to give us the minimum amount of tension, the amount of the ability to, if that's 100% of our maximum that muscle can do. You come down here and decrease the length to a certain amount. For example, there's no place for that to go. I mean, you're not going to be able to generate any force. So a little bit of stretch is going to increase this guy to maybe right here. And you come to this area, and when you get there, you have got the maximum amount of force that you can generate. But you can go too far on this too. There's our actin. There's our myosin. You stretched it too far. These guys can't even make contact with the actin, so there's going to be no force. So you see that there's an optimum amount of stretch on a muscle that'll give you the most amount of force. But if you continue to stretch it, you get to the point where you absolutely, right here, there are no cross bridges. There's no way. See, this one, you have the actin actually overlapping there. Here, you've got a good cross bridge, but you can probably do better than that. Anyway, that's the length and tension relationship. The longer the sarcomere is stretched to a point, right there, to be specific, the better and the more force you're going to be able to generate. If you stretch it too far, though, you lose it. Okay, latency, contraction, and relaxation. So the neuron fires and releases it, but I don't actually get a contraction instantly. That's like 5 milliseconds there, guys. Why don't I? Because it takes time for the action potential to actually, it takes time for the neurotransmitter to float across the synaptic cleft. It takes time to bind. It takes time for the action potential, the spread of the voltage, to travel over the sarcolemma. That's the latent period. It's just going to take a certain amount of time. It's not instantaneous. So that's our latent period. Contraction period, you see, we start and we gradually build up until we get to the maximum that we're doing there. And then, and this starts from like 5 to about 25, a little over 25 milliseconds. A millisecond is 1/1000th of a second, guys. No, it's not instantaneous. It actually is a finite amount of time. But check this out. From the time that this guy starts to relax, it takes him a long time to come all the way back down. There's a long relaxation period. But remember, what we're doing too is taking that calcium and putting it back into the SR. And so this takes a little bit of time. Okay, here's some summation of wave and tetanus. So tetanus is real easy. So this is a contraction we saw. We come here and we just kind of stay contracted. What would do that? Like a Charlie horse, like you might get in your calf muscle. It's a sustained contraction. And that's what the disease tetanus is named after. Lockjaw is a sustained contraction. Now, wave summation, what's happening here is this guy is contracting. He starts to relax. Remember, it's a long relaxation period. And before he can go very far, he gets another nerve signal. So he contracts a little bit more, starts to relax. Before he gets very far, another nerve signal comes through. That's wave summation. So what we're doing is actually adding up. These guys are not getting a chance to go all the way back down to 0 before the next nerve impulse. So they're kind of stepping their way up. This is called trepe. One thing I want you to notice about this is that each of these is slightly higher. So what this is over a period of time, the muscle gradually kind of stair steps its way up to increased force. And that's going to be our trepe. Muscle tone. The only time you have no muscle tone is when you're dead. You're going to have even partial contractions, even when you're asleep. This helps to keep our joints together. It pulls the bones together, especially on the shoulder joint. There's not much in the way. I mean, the ligaments are there, and that's great. And there's a little bit of a capsule, not much of a joint on the glenoid cavity. So it's actually the contraction of the rotator cuff muscles that hold the arm into the shoulder joint. We also use these guys to just stand up straight to maintain our posture, the erector spinae group of muscles. When we get damage to our central nervous system, that could be the spinal cord, spine injury, and we reduce the nerve impulses, that can actually cause the muscles to become hypo-responsive or even totally paralyzed, and in which case the muscles will actually start to go away. If a muscle does not get nerve impulses telling it to contract, it will eventually go away. It's going to atrophy. Hypertonia is just the opposite. Excessive tone, and usually you get exaggerated reflexes as well. Types of muscle fibers, let's divide these up into their three. Now, these muscle fibers are distinct as far as their function and what they can and can't do. But this is something that you probably should know, is that these muscle fibers can change from one type to another depending on what you do with your muscles, what type of exercise or activity you do. Okay, we have the slow oxidative fibers. The key word there is oxidative. These guys are going to use the mitochondria, aerobic metabolism. They have lots of mitochondria. These guys are going to be relatively small diameter muscle fibers. They're going to have lower tension. These guys are going to have a big blood supply because they need oxygen, and they're going to look kind of reddish in color. These guys are muscles that you can keep going for a fairly decent amount of time. So they're resistant to fatigue. They don't give out very quickly. Okay, I'm going to talk about the fast glycolytic next. The fast glycolytic, these guys use glycolysis. They're not about the mitochondria. They are large in diameter. They're going to have lots of glycogen, and these guys are going to be capable of significant force generation. They don't have those mitochondria or the myoglobulin. Myoglobulin, I didn't tell you about that. We haven't talked about it yet, but myoglobulin is, you know how hemoglobin in the red cells carries oxygen? Myoglobulin is a storage mechanism for oxygen inside the muscles. So in addition to storing their own sugar, they also store some oxygen. So these guys don't have a lot of myoglobulin, and these guys are going to be wine in color. The fast fibers are used for quick, forceful contractions and powerful movements. The trade-off is these guys give out very quickly, so they only can be used for a relatively short period of time. So they're fast, but they don't have any endurance. These guys are slow, but they have endurance. The fast oxidative, they use the anaerobic metabolism, but they're somewhere in between these two guys. That's why I wanted to save them for last. Exercise, you can do resistance exercise, which is what gives hypertrophy, the buildup of muscle. Or you can do endurance, which is like the cardio. These guys are going to be for the slow oxidative. These guys are going to be for the fast glycolytic. Cardiac muscle, we're going to talk more about that in anatomy and physiology too. They're striped, but these guys are branched. You can see this area here, that's the gap junction. They have desmosomes that hold these together, but they also have these gap junctions which allow those sodium ions to travel through from one cell to another really quickly. That's the intercalated disc right there, which joins these muscle cells together. That's really all we're going to talk about cardiac. You will pick it up later on in the next course. Smooth muscle. Smooth muscle is world champion when it comes to being able to be fatigue resistant. These guys can hold on for a while. They're different. They're kind of fusiform shape, which is kind of like that. Have kind of cigar-shaped nuclei. These guys have nerves kind of come in like that, but they don't have a motor end plate per se. So these guys also have something called a dense body and these intermediate filaments. And when these guys contract, it's almost like they kind of corkscrew like that. You can see that they don't just do a straight, get shorter. Here's the thing, these are called varicosities. These guys don't have a motor end plate, or, well, they don't have a neuromuscular junction. These guys, these are neurons, and those are vesicles with neurotransmitter. These are also autonomic nervous system. This is an unconscious control. And these guys kind of synapse with these guys. And by synapse, I mean they come within a short distance. Still going to have that gap. Atrophy is what happens. So this is extreme, but even in the 6 weeks in which you have an arm in a cast, you will see some loss of muscle mass as well as the bone mass, but simply because it's not being used. Hypertrophy, there's two things you can get. You get hypertrophy and hyperplasia. Now, hyperplasia is when you make more cells. Hypertrophy is when the cells you have get bigger. All right, guys, that's got it for Chapter 10 Muscle. I'll see you on the next one.